Hello student, welcome to EPG Pathsala. I am Dr. Asutosh Rai from ICR, Indian Institute of Vegetable Research. Today we are going to talk on module single stranded DNA vectors, M13, FD and F1, yeast artificial chromosomes, bacterial artificial chromosomes, P1 derived artificial chromosomes and binary bacterial artificial chromosomes under the paper Genetic Engineering and Recombinant DNA Technology. After completing this module, you should be able to understand single DNA vectors like M13, FD and F1, yeast artificial chromosomes, bacterial artificial chromosomes and binary bacterial artificial chromosomes and P1 derived artificial chromosomes. Phage vectors are plasmids having a small segment of filamentous phage M13, FD or F1 phage. These are capable to carry up to 10 kb of passenger DNA. Examples are P-EMBL series of plasmid vectors and P-blue script family plasmid vectors. The M13 origin of replication allows the packaging of plasmid into a M13 phage when the bacteria is also infected with M13 helper phages. Plasmid generally encode no or one kind of coat proteins. Other structural and functional proteins necessary to accomplish the life cycle phagmids are encoded by life cycle of phagmids are encoded by helper phage and generally transcribed by host. Replication in M13 bacteriophage. In M13 bacteriophage, DNA is replicated by a rolling circle mechanism. In this mechanism, one strand is nicked and the free 3' OH is extended by DNA polymerase. The 3' end on the circle is extended while the growing point rolls around the circle template. The 5' end is displaced and forms a tail of single-stranded DNA. The single-stranded tail is converted into double-stranded DNA by synthesis involving RNA primers. In present table, we can see different types of cloning vectors having their compatible host, insert size, origin of replication, and their form of DNA. Here you can see M13 vectors which have E. coli host, and these can accommodate 1 to 4 kb DNA. These are transducted, these have F1 origin of replication, and they are in circular form present in host cell. Likewise, lambda phage, P1 derived artificial chromosomes, bacterial artificial chromosomes, and yeast artificial chromosomes, the different features you can see in the table. The members of the FF class of bacteriophage, that is M13, FD and F1, have been used extensively as vectors for recombinant DNA cloning. These filamentous male specific coli phages have two important features that aid in their use as cloning vectors. They do not lyse their host and they allow packaging DNA that is longer than phage unit length. However, perhaps the most significant advantage of this group of phages is that they replicate in double-stranded supercoiled DNA but package only circular single-stranded molecule. Consequently, single-stranded DNA can be prepared quickly and in sufficient purity directly from phage particles. Therefore, these vectors are particularly well studied for rapid sequence analysis by Sanger deoxy chain termination method and have been used extensively in oligonucleotide direct mutagenesis. FD bacteriophages. These infect only negative bacteria bearing F episome. These are filamentous bacteriophage. FD is type of filamentous bacteriophage known to infect Escherichia coli. It shares many structural and genomic similarities with Enterobacter phage M13. Phage FD engineered to display immunogenic peptides may be useful as vaccines. F1 bacteriophages. Bacteriophage F1 is structurally classified as a class 1 filamentous phage and is used related to the other FF phages such as M13 and FD. F1 is a filamentous rod-shaped single-stranded DNA phage with a molecular mass of about 1.6 into 110 Daltons. By weight, it is 11.3% DNA. The thousands of identical major code proteins that make up the sheath and arranged in a fascicle-like alpha helix. The ends of filament are sealed with protein caps. The blunt end contains 3 to 5 copies of each 7 and ninth class of proteins, while the terminally extruded bead-like ends contain about 5 copies of each 3 and 6. F1 has a circular 6407 base pair genome of single-stranded DNA. There are 11 genes encoded on the genome. 2 are overlapping in frame genes. 5 of the encoded proteins make up the viron. 3 are needed for the synthesis and the rest are for assembly. The genes are generally referred by Roman numerals first to 11th and are in order second X, 5, 7, 9, 8, 3, 6th, 1 and 11 and 4th intergenic regions that are IG or IGR regions. The IGR contains the packaging signals that are PS as well as sequences that dictate termination, nicking for replication and the binding of second and IHF in F1 phages. F1 is male specific, it only infects HFR or HF plus strains of E. coli. Bacteriophage M13 phage is filamentous phage that infects E. coli via F. pilus. The genome is a single-stranded circular DNA of size approx 6.4 kb, surrounded by a proteinaceous coat called capsid. The DNA strand present in phage is called plus strand. After entering E. coli host, it converts into double-stranded DNA molecule called replicative form, that is RF, by utilizing bacterial machinery. M13 phages are used as cloning vector, can be contained in both single stranded as well as double stranded form. Replicative form double stranded vectors are modified and replicated inside E. coli hosts similar to the plasmid vectors. Single stranded vectors can be isolated by collecting M13 phages for sequencing. High cloning capacity vectors. High cloning capacity vectors are generally used for construction of genomic libraries. These are cosmids, bacterial artificial chromosomes, P1 derived chromosomes, and yeast artificial chromosomes. They are designed to handle longer DNA insert, much larger than the lambda replacement vectors. So they require lower number of recombinants to be screened for identification of a particular gene of interest. Artificial chromosomes are DNA molecules assembled in vitro from defined constituents that can function like natural chromosomes. Types of artificial chromosomes, when we discuss about types, then first comes P1 derived artificial chromosomes, then bacterial artificial chromosomes, binary bacterial artificial chromosomes, yeast artificial chromosomes, human artificial chromosomes, and nowadays mammalian artificial chromosomes. So before talking about P1 derived artificial chromosomes, we should have a look on bacteriophage P1. The bacteriophage P1 is a template phage that infects E. coli. In its lysogenic cycle, the P1 genome remains as a plasmid in bacterium. One of its important feature is that it hijacks the host machinery and integrates it into host genome. The viron P1 has isohedral head and a tail with 6 fibers which help it to anchor the host cell wall. The P1 phage have a comparatively large genome, approx 93 kilobase, as a linear double-stranded DNA molecule. After insertion into the host, it circularized and replicates as plasmid. The P1 has two ORIs, origin of replication, that ORI are responsible for lysogenic cycle, whereas a ORI L replicates it during lytic cycle. It can carry a foreign DNA up to 100 kilobase pair, and it is able to replicate into the host cytoplasm. M13 phages or M13 based bacteriophages replicates in a rolling circle DNA replication mechanism. The rolling circle DNA replication is initiated by an initiator protein encoded by the bacteriophage DNA. This nicks one strand of double-stranded circular DNA molecule at a site called double strand origin or DSO. The initiator protein remains bound to 5' phosphate end of the nick strand, and the 3' hydroxyl end is released to serve as primer for DNA synthesis by DNA polymerase III. Using the unnicked strand as a template, replication proceeds around the circular DNA molecule, displacing the nicked strand as a single-stranded DNA. The displacement of nicked strand is carried out by a host-encoded helicase in the presence of replication initiation protein. Continued DNA synthesis can produce multiple single-stranded linear copies of original DNA in continuous head-to-tail series called concatamers. These linear copies can be converted to double-stranded circular molecules through the following process. First, initiator protein makes another nick to terminate synthesis of the first leading strand. RNA polymerase and DNA polymerase III then replicate the single-stranded origin, that is SSO DNA, to another double-stranded circle. DNA polymerase III removes the primer, replacing it with DNA, and the DNA ligase joins the ends to make another molecule of double-stranded circular DNA. By this way, plasmid has certain advantages. Their carrying capacity of plasmid is higher than phage vectors. Plasmids has higher efficiency in transformation than phage vectors, and plasmids are genetically more stable than recombinant phage vectors. Plasmids can be exploited to generate single-stranded DNA templates for sequencing purposes. Single-stranded plasmid vectors inside the phage can targeted for site-directed mutagenesis also. These single-stranded vectors can be used to generate hybridization probes for mRNA or cDNA. One of the first hybrid plasmid vector was PEMBL, constructed in 1983. They are characterized by the presence of a BLA gene as selectable marker for ampicillin resistance, a short segment coding for alpha peptide of beta galactosidase LAC-Z and containing an MCS, that is multiple cloning site. Third, the intergenic region IG region of phase 1 for replication of origin. These vectors have been used successfully for DNA sequencing with the dideoxy method and can be used for other purposes for which M13 derivatives are used. However, the PEMBL plasmids have the advantages of being smaller than M13 vectors, and the purification of DNA is simple. In addition, long inserts have a higher stability in PEMBL plasmids than M13 vectors. Within the bacteriophage, such as M13, the replication process is complex. It is in phage DNA molecule generally carry several genes essential for replication, including genes for components, phage code proteins, and phage specific DNA replicative enzymes. Alteration in any of these genes will impair or destroy the replicative ability. So, there is less freedom to modify phage DNA molecules. For the in vitro packaging of phage particles, plasmid vectors like PMBL8 is used a helper phage. Here in figure, you can see the whole scheme of replication in plasmid vectors and how new virions are created. The PMBL8 was made by transferring 1300 base pair fragment of M13 into PUC8 vectors. This piece of M13 in PMBL8 contains single sequence recognized by enzyme that converts the normal double stranded M13 molecule into single stranded DNA before secretion of new phage particles. The single sequence still remains functional even though detached from rest of M13 genome. When a normal M13 is used as helper phage, this provides necessary replicative enzymes and phage coat proteins. Bacterial artificial chromosomes are designed for cloning of large DNA insert, typically approx 100 to 100 to 300 kilobase in E. coli host. Bacterial vectors contain a single copy of F plasmid, origin for replication or ORI. The F fertility plasmid is relatively large and vectors derived from it have a higher capacity than normal plasmid vectors. F plasmids have fertility factor which controls the replication and maintains a low copy number. Also, conjugation can take place between F plus bacteria, that is male, and F minus bacteria, that is female, to transfer the F plasmid via pilus. P1 derived artificial chromosomes are constructed by combining the best features of P1 bacteriophage vectors and BAC, that is bacterial artificial chromosomes. No chimeras or instability is associated with this vector and it is used for genomic mapping. It is introduced by electroporation and the carrying capacity of P1 derived artificial chromosomes is about 60 kilobase pairs to 150 kilobase pairs. Thus, these are E. coli based vectors, are low copy number, stable for maintenance of foreign DNA, and capable of having 150 kilobase pair of foreign DNA. Bacterial artificial chromosomes, that is BAC. It was developed to prepare a larger insert clone library of genome sequencing project and was first used in human genome project. The cloning capacity of BAC vector is up to 300 kilobase pair. It gained popularity in genome sequencing project due to its high cloning capacity. It is maintained as a single copy plasmid It is characterized by stable maintenance even after more than 100 generations. Its origin of replication is F plasmid. It has three genes of partitioning such as par A, par B, and par C. The host bacteria should be deficient in homologous recombination and it should be Rec A. The size of vector, that is bacterial artificial chromosome vector, is between 7.4 kilobase pair. After ligating with the foreign DNA, recombinant bacterial artificial chromosome is introduced into E. coli by electroporation. In figure, you can see different parts which are necessary for a bacterial artificial chromosomes. These segments are par C, par B, par A, Rep E, ampicillin resistant gene, LOXP as selectable marker, multiple cloning sites, and COS-N region. Each artificial chromosomes. First, it was described in 1983 by Murray and Jostak. Each artificial chromosome has sequence to exist inside E. coli as a circular plasmid and contains a sequence to maintain it as linear nuclear chromosome in yeast. The number of clones in genomic libraries can be greatly reduced with the use of yeast artificial chromosomes. Yeast artificial chromosome have following elements. First, it has a E. coli origin of replication, it has yeast origin of replication, it has elements of eukaryote yeast chromosomes, that is first centromere and telomeric region. The selection markers for both the host bacteria as well as yeast. Yeast artificial chromosomes can be maintained as a linear DNA chromosome. It can be introduced into the yeast cell by electroporation. Yeast is a vector used to clone DNA fragment larger than 100 kb up to 3000 kb. Yeast artificial chromosomes are also useful for physical mapping of complex genomes and for cloning of large DNA fragments. If we talk about yeast artificial chromosome, it contains the centromere region and telomere region and an autonomous origin of replication, that is ARS. This is responsible for replication in yeast. This element which are required for replication and conservation of yeast artificial chromosome in yeast. ARS elements are thought to be act as replication of origin. A yeast artificial chromosome is built using an initial circular plasmid which is typically broken into two linear molecules using restriction enzyme. Then the DNA ligase is used to ligate a sequence or gene of interest between these two linear molecules, forming a simple larger linear piece of DNA. The TRP1 and URA3 genes are included in yeast artificial chromosome vector to provide a selection system for identifying transformed yeast cells that introduced into yeast artificial chromosome by complementing recessive allele TRP1 and URA3 in yeast host cell. Bacterial artificial chromosomes are bacterial cloning vectors derived from F plasmid that can accommodate up to 350 kilobase pair, mostly commonly 120 to 150 kilobase pair of DNA sequence. This has a considerably lower error rate than the still larger capacity yeast artificial chromosomes. Bacterial artificial chromosomes are usually exist in single copy per cell. Random bacterial artificial chromosomes are selected at random from genomic library and are then shotgun sequenced. Most bacterial artificial chromosome vectors lack selectable marker suitable for mammalian cell selection, but can be retrofitted by employing the CRE or LOXP sites for a specific recombination system. Another class of bacterial artificial chromosomes are binary bacterial artificial chromosomes. The development of bacterial artificial chromosome system should greatly reduce the time and effort involved in the gene verification step and allow for rapid introduction of valuable agronomic traits into unrelated plant species. The binary bacterial artificial chromosomes may make it possible to express a series of genes or entire biochemical pathway from any organism in plant system. These are potentially produced for pharmaceuticals or for novel materials in plants. The binary bacterial artificial chromosomes BIBAC vectors combine two technologies. First is bacterial artificial chromosome and the binary vector strategy for agrobacterium mediated plant transformation. The binary bacterial artificial chromosome vector is suitable for bacterial host E. coli and Agrobacterium tumefaciens and is capable of maintaining at least 150 kilobase pair of foreign eukaryotic DNA into E. coli and also in Agrobacterium tumefaciens. Binary bacterial artificial chromosomes has a unique BAM-H cloning site and is suitable for construction of genomic DNA libraries. In addition, these are two pairs of restriction sites, not one, and SFI1 that flanked by BAM-H1 site. The virulent helper plasmid that were designed to be used in conjugation with binary bacterial artificial chromosome vector to enhance the transfer of high molecular weight DNA into the plants. So in this module, we have learned about single standard bacteriophages that are FF, FD and F1 and M13 vectors, plasmid vectors, replication in these vectors and high cloning capacity vectors like bacterial artificial chromosome, binary bacterial artificial chromosomes and P1 derived artificial chromosomes. Thank you.